Welcome to the deep dive. You ever stop and think about how life manages to stay so incredibly organized? The universe is always leaning towards chaos, right?
It really is. It's a constant battle.
And the answer, well, a huge part of it, boils down to this one molecule, ATP. It's like the cell's energy cache.
That's exactly it. And the power plants making most of that cash in our cells, well, in eukaryotic cells anyway, are these amazing little organelles. Mitochondria and chloroplasts. One burns fuel, the other catches sunlight.
But they're surprisingly similar in some ways, aren't they? Evolutionarily speaking, and even how they work fundamentally.
Absolutely. And that's what we're digging into today. We're doing a deep dive using molecular biology of the cell, 7th ed, as our guide to really explore these cellular powerhouses.
Yeah, our mission here is to peel back the layers, look at the nuts and bolts, the molecular machines, and figure out the why and how of cellular energy. We want to make this complex stuff, stick.
And the core concept, the thing that ties so much of this together, is something called chemiosmotic coupling. It sounds technical, but it's this beautiful central idea.
Okay, chemiosmotic coupling. Break that down for us.
Well, it's basically a direct link between making chemical bonds like the ones in ATP and moving stuff across membranes. And it's ancient. Bacteria do it, mitochondria do it, chloroplasts do it.
Right. So how does it work, like step by step?
Okay, 2 main stages. First, you get these high-energy electrons. They might come from food you ate or maybe from sunlight hitting a pigment. These electrons get passed down a chain, an electron transport chain. Think of it like a bucket brigade, but with electrons and proteins embedded in a membrane.
And as they get passed along?
Energy is released, little controlled bursts, and that energy does something really important. It pumps protons, just hydrogen ions, H+, across that membrane.
Okay, so you're building up protons on one side, like charging a battery.
Exactly. You create this electrochemical gradient. More protons on one side, fewer on the other, plus a voltage difference. It's stored energy, what we call the proton motive force.
Got it. Stage 1, build the gradient. What's stage 2?
Stage 2 is tapping into that energy. The protons want to flow back down their gradient, and they do, but they have to go through a specific channel.
And that channel is?
An amazing ATP synthase enzyme. It's like a tiny molecular water wheel or a turbine. The flow of protons spins part of it.
It actually spins.
Oh, yeah. It physically rotates. And that rotation, that mechanical energy, is directly used to smash ADP and phosphate together to make ATP. It's an incredible piece of nanomachinery.
Wow. So proton flow spins a turbine that makes ATP. That's chemiosmosis in a nutshell.
Pretty much. And it's remarkably efficient. It captures almost half the energy released from those electrons. Way better than just burning fuel uncontrollably. It's how cells fight that chaos you mentioned.
Okay, let's zoom in on mitochondria first. The cell's powerhouse, right? But they're not just static beings.
Not at all. They're super dynamic. They can make up like 20% of a cell's volume, constantly moving, changing shape, dividing, fusing together. It's a whole network.
And they hang out where the action is, don't they?
Absolutely. Location, location, location. In heart muscle, packed right next to the contracting fibers, sperm tail, wrapped around it for power, neurons. They can travel down long axons on microtubule tracks to reach synapses that need energy.
Yeah, that makes sense, fueling the demand. And they even interact with other parts of the cell, like the ER.
Yeah, they have these dynamic contacts with the endoplasmic reticulum. They swap lipids, calcium ions. These contact points are even involved in helping mitochondria divide. It's all interconnected.
Looking at the big picture, getting mitochondria was a game changer for evolution, wasn't it?
Oh, hugely pivotal. Think about basic energy production like glycolysis. You get maybe 2 ATP per glucose molecule. That's it. Not much at all. But with mitochondria fully breaking down that glucose, you get about 30 ATP, a 15-fold increase. That massive energy boost is likely what allowed complex, multicellular life like us to evolve.
So let's look inside the structure. You said 2 membranes.
Right. An outer membrane that's pretty leaky to small stuff because of proteins called porins. So the space between the membranes, the inner membrane space, is chemically similar to the main cell cytoplasm.
But the inner membrane is different.
Very different, much less permeable. And it's folded like crazy. These deep folds are called cristae.
Why all the folds?
Surface area. It massively increases the area for the electron transport chain and ATP synthase. In a heart cell, it can be 20 times the area of the cell's outer membrane. If you laid out all the cristae in your body, it'd cover a football field. Seriously.
A football field inside us. That's wild.
It's where the energy conversion happens. Inside the inner membrane is the central compartment, the matrix.
And you mentioned they divide and fuse.
Yeah, there's this constant balancing act. Fission, splitting apart, and fusion, joining together. It controls their shape, number, and where they go.
How does that work?
Fission uses specific proteins related to dynamine that kind of pinch and cut both membranes. Fusion is more complex, separate machines for the outer and inner membranes.
What happens if one gets damaged?
The cell has a cleanup crew. It's called mitophagy, targeted self-eating of mitochondria. If a mitochondrion loses its proton gradient, it gets tagged for destruction by proteins like PINK1 and parkin.
Yeah.
Problems with the system are linked to Parkinson's disease, actually.
Interesting connection. Okay, the matrix, the innermost part, what goes on in there?
That's the main metabolic hub. It takes in fuel, mostly pyruvate from sugars and fatty acids from fats.
And does what with them?
Breaks them down further. The big pathway here is the citric acid cycle, or Krebs cycle. It takes acetyl-CoA, made from those fuels, oxidizes it, releases CO2.
The stuff we breathe out.
Exactly. But the crucial product is high-energy electrons carried by molecules like NADH.
And that NADH.
Goes straight to the electron transport chain on that inner membrane we talked about. That's the direct link from food breakdown to ATP synthesis.
But mitochondria do more than just ATP, right?
Oh, absolutely. They're involved in regenerating other essential molecules, providing building blocks for amino acids, making for hemoglobin, making iron-sulfur clusters.
Iron-sulfur clusters.
Yeah, critical components for lots of proteins, including some in the electron transport chain itself, and even for maintaining the nuclear genome. They also make unique lipids like cardiolipin, which helps shape the cristae.
And signaling, too.
Yep. Buffering calcium, generating some reactive oxygen species as signals, even regulating programs, cell death, apoptosis. They're way more than just power plants. They're central metabolic and signaling hubs.
Okay, let's circle back to that chemiosmosis, the electron transport chain, pumping protons, this whole oxidative phosphorylation thing.
Right. So we said energy is released as electrons move from NADH ultimately to oxygen. It's like burning hydrogen and oxygen, which is explosive.
But the cell does it gently.
Exactly, stepwise. Controlled release. Capturing energy efficiently instead of losing it all as heat.
And storing it as that electrochemical gradient. 2 parts, you said.
Yeah. A difference in proton concentration, the pH gradient, the matrix becomes alkaline around pH 8, and a voltage difference across the inner membrane, the matrix side becomes negative.
And together that's the proton motive force.
That's it. About 180 millivolts usually. A strong driving force pulling protons back into the matrix. It comes down to redox potential, how much a molecule wants electrons. NADH is a weak holder, oxygen is a strong grabber. That difference drives the whole thing.
Who are the electron carriers again?
You've got proteins with metal ions, like iron or copper.
Yeah.
Cytochromes with heme groups, those iron-containing rings.
Yeah.
Iron-sulfur proteins. And this little mobile carrier, ubiquinone or a coenzyme Q, shuttling electrons within the membrane.
And they work in teams, these big complexes.
Right. Usually 4 main complexes in the inner membrane, sometimes even grouped together as supercomplexes for efficiency.
Tell me about them. Complex I.
NADH dehydrogenase, it's the biggest, takes electrons from NADH, passes them through various carriers to ubiquinone, and crucially, it pumps protons about 4H plus for every pair of electrons, starts building that gradient.
Okay, complex 3.
Cytochrome C reductase takes electrons from ubiquinol, well, its reduced form, ubiquinol, and passes them to another mobile carrier, cytochrome C. It uses a really clever mechanism called the Q cycle to pump more protons across the membrane.
And complex 4, the end of the line.
Cytochrome C oxidase takes electrons from cytochrome C, and this is critical, passes them to molecular oxygen, O2.
The oxygen we breathe in.
Precisely, and it does it safely. It holds onto the oxygen molecule until it has collected 4 electrons, then releases it as 2 harmless water molecules. This prevents nasty reactive oxygen species from forming. It accounts for like 90% of our oxygen use.
Is that why cyanide is so bad? It blocks this?
Exactly. Shuts down complex 4, electron transport stops, ATP production plummets. Fatal.
You mentioned 4 complexes.
Okay.
What about complex 2?
Complex 2, succinate dehydrogenase. It's actually part of the citric acid cycle, too. It feeds electrons from a different carrier, FADH₂, into the ubiquinone pool. But the key difference, it doesn't pump protons.
I see. And how do protons even get through these big protein complexes?
It's thought they use these proton wires chains of amino acid side chains or water molecules that allow protons to kind of hop rapidly through the protein structure, linked to conformational changes driven by electron transfer.
Okay, so the gradient is built. Now the payoff. ATP synthase, the turbine.
The amazing ATP synthase. It's an F-type ATPase. And the scale is just mind-boggling. You mentioned we turn over kilograms of ATP a day. This machine makes almost all of it from glucose oxidation, roughly 30 ATP per glucose.
Why so much ATP? Why is it so important?
Because ATP hydrolysis releases a lot of free energy. Cells keep the ATP level really high compared to ADP. This makes ATP hydrolysis energetically very favorable, allowing it to drive countless other reactions in the cell that wouldn't happen on their own. Keeps everything running.
So the turbine mechanism again. Protons flow through.
Through the membrane part, the faux part. It's a ring of subunits, usually C subunits. Proton flow makes this ring spin.
Spin relative to what?
Relative to the head part, the F1 ATPase, which sticks out into the matrix. The spinning ring turns a central stalk, the gamma subunit, inside the stationary F1 head.
Like a camshaft.
Exactly like a camshaft. As the gamma stalk rotates, it bumps against the subunits in the F1 head, the alpha and beta subunits, forcing them to change shape.
And these shape changes make ATP.
Yes, one shape binds ADP and phosphate. The rotation pushes it into another shape that forces them together to make ATP. Then another shape change releases the ATP. 3 ATP is made for every full rotation, and it spins fast.
How fast?
Maybe 8,000 RPM, churning out hundreds of ATP molecules per second per single enzyme. It's converting proton flow into rotary motion into chemical bond energy, direct mechanical to chemical conversion.
That's incredible. Is this machine unique to mitochondria?
Nope. It's ancient. Found in bacteria, chloroplasts too, all using a proton gradient. The number of C subunits in the rotor can vary, which changes how many protons it takes per ATP and evolutionary tuning.
And it can run backward.
Yeah, if needed. It can hydrolyze ATP to pump protons out. Sometimes bacteria use this to maintain their gradient for other things, like transport or spinning their flagellar.
You mentioned the cristae folds before. Does ATP synthase play a role there?
It does. ATP synthase actually forms dimers, pairs that line up along the sharp ridges of the cristae. These rows of dimers help bend the membrane to create those folds.
Wow, so they shape their own environment.
And this arrangement seems to create a sort of proton trap near the ATP synthase, funneling protons efficiently to the enzyme even if the overall gradient isn't huge. Very clever design.
What about getting ATP out and ADP in?
Special carrier proteins in the inner membrane handle that. The main one is the ADP-ATP antiporter. It swaps one ATP out for one ADP in, driven by the voltage component of the proton gradient, keeps the ATP factory supplied, and delivers the product.
Is there any way to bypass this? Make heat instead?
There is. Brown fat cells and beige fat cells have an uncoupling protein. It just lets protons leak back into the matrix without going through ATP synthase. The energy is released as heat. That's how babies and hibernating animals stay warm. It's being studied for obesity, too.
So chemiosmosis is fundamental, even bacteria.
Absolutely. Bacteria were doing it long before mitochondria existed. They used proton gradients across their plasma membrane for ATP synthesis, nutrient import, flagellar rotation. Some even use sodium gradients. The principle is universal.
How did it evolve?
The thinking is it started with simple pumps, maybe ATT-driven, to control internal pH. Then came electron transport-driven pumps using environmental energy sources.
Right.
Finally, coupling these proton pumps to ATP synthase created this highly efficient energy system.
Okay, shifting gears, let's talk chloroplasts, the solar panels.
Right, photosynthesis, using sunlight, water, and CO2 to make sugars and oxygen, the foundation of most ecosystems.
And they produced the oxygen in our atmosphere originally.
Well, their ancestors did. Cyanobacteria. Billions of years ago, they started releasing oxygen as a byproduct of photosynthesis. Changed the planet forever, paving the way for aerobic life. A monumental event.
How are chloroplasts structured? Similar to mitochondria?
Some similarities, they're generally larger. Got an outer and inner membrane, the envelope. Inside is the stroma, like the mitochondrial matrix. That's where ATP is used for carbon fixation. It also has its own DNA and ribosomes.
But there's an extra membrane system.
Yes, the thylakoid membrane. It's a third system inside the stroma, forming flattened sacs called thylakoids, often stacked into grana. This is where the light-capturing machinery, electron transport, and ATP synthase are located in chloroplasts. The space inside the thylakoids is the thylakoid lumen.
So photosynthesis has two parts.
Broadly, yes.
Yeah.
Stage 1, the light-dependent reactions in the thylakoid membrane. Light energy is captured, used to split water, release oxygen, pump protons into the thylakoid lumen, creating a gradient there, and make ATP and NADPH in the stroma.
Okay, light capture makes ATP and NADPH. Stage 2.
The carbon fixation reactions, sometimes called the Calvin cycle or light-independent reactions, these happen in the stroma. They use the ATP and NADPH from stage 1 to take CO2 from the air and convert it into sugars.
How does that CO2 capture work?
The key enzyme is rubisco. It attaches CO2 to a 5-carbon sugar, ribulose 1,5-disphosphate. Rubisco is incredibly important and maybe the most abundant protein on Earth.
Why so abundant?
Partly because it's actually pretty slow, and it has a problem. It can mistakenly grab oxygen instead of CO2, especially when it's hot and dry.
What happens then?
That leads to photorespiration, which wastes energy and releases already fixed carbon. Not good for the plant.
But some plants avoid this.
Yeah, C4 plants like corn and sugarcane have evolved a workaround. They use a preliminary step to concentrate CO2 in specialized cells around rubisco. It costs extra ATP, but it minimizes photorespiration, giving them an edge in hot climates. Clever adaptation.
What happens to the sugars made?
They can be used immediately, stored temporarily as starch inside the chloroplast, or converted to sucrose for transport to other parts of the plant. At night, stored starch is broken down to fuel the plant's own respiration in its mitochondria, just like animals use stored fat.
How is light actually captured? By chlorophyll.
Mostly, yes. Chlorophyll absorbs red and blue light, gets an electron excited, but most chlorophyll isn't directly involved in energy conversion. They're in antenna complexes. They gather light energy and funnel it, like resonance energy transfer, from molecule to molecule until it reaches a reaction center. There, a special pair of chlorophylls actually gives up an electron, converting light energy to chemical energy.
And there are 2 systems doing this.
Right. Photosystem 2 and photosystem I, working in series. It's called the Z scheme because of how the electron energy levels look on a diagram.
What does photosystem 2 do?
It does something unique and amazing. It splits water molecules to get electrons. It uses a cluster of manganese atoms. This is where the oxygen we breathe comes from. And the protons released help build the gradient inside the thylakoid lumen.
Okay, electrons from water go through PSII. Then where?
To a cytochrome B6F complex, very similar to complex 3 in mitochondria. It links the 2 photosystems and pumps more protons into the thylakoid lumen using a Q cycle, boosting that gradient.
And photosystem Y.
It takes the electrons, gets them re-energized by another photon of light, and passes them ultimately to make NADPH, that other key energy carrier needed for carbon fixation.
So chloroplasts also make ATP using a proton gradient.
Yes, using a very similar ATP synthase. Protons flow out of the thylakoid lumen, where they were pumped, back into the stroma through ATP synthase, making ATP in the stroma. The gradient here is mostly pH-based, a huge difference across the thylakoid membrane.
Can they adjust the ratio of ATP and NADPH they make?
They can. Photosystem I can run in a cyclic mode, sending electrons back to the cytochrome complex instead of making NADPH. This just pumps protons and makes more ATP. It lets the chloroplast fine-tune energy production based on the cell's needs.
This whole system, mitochondria and chloroplasts, points to endosymbiosis, right? Cells engulfing bacteria.
The evidence is overwhelming. Mitochondria look like descendants of aerobic alpha proteobacteria. Chloroplasts, like descendants of cyanobacteria, engulfed billions of years ago and became permanent residents.
And they still have their own DNA.
They do. Both have their own small, usually circular genomes, plus ribosomes and the machinery to make some of their own proteins. It looks very prokaryotic inside.
But most of their proteins come from the nucleus.
The vast majority, yeah. Encoded in the cell's nuclear DNA, made in the cytoplasm, and then imported into the organelle using specific targeting signals. There's been massive gene transfer from the organelles to the nucleus over evolutionary time.
Does the mitochondrial DNA have any weird features?
Human mitochondrial DNA is super compact, almost no junk DNA. It uses fewer types of tRNA molecules because the codon rules are a bit relaxed. And crucially, the genetic code itself is slightly different from the universal code. UGA means tryptophan, not stop, for example.
Why the different code?
Probably just random genetic drift in these small, isolated genomes.
Do proteins ever go the other way? Out of mitochondria?
Rarely. The main example is cytochrome C being released to trigger apoptosis, programmed cell death.
How do these organelles divide? Still like bacteria?
Chloroplasts, mostly yes, using bacterial-like FTSE proteins. Mitochondria, interestingly, use dynamin-related proteins, more like eukaryotic systems for membrane fission, shows some divergence after endosymbiosis.
And how are they inherited? We get them from?
Mostly from your mother. In animals and plants, it's predominantly maternal inheritance. The egg cell contributes almost all the cytoplasm, including the mitochondria. Sperm mitochondria usually get destroyed after fertilization.
Does this inheritance pattern matter?
It does for mitochondrial diseases. Mitochondrial DNA mutates faster than nuclear DNA, partly due to reactive oxygen species nearby and less robust repair.
Why?
If harmful mutations accumulate, they're passed down maternally. Because cells have many mitochondria and they segregate randomly when cells divide, the severity of these diseases can vary hugely depending on which tissues get more faulty mitochondria. Often affects high-energy tissues like muscle and brain.
That random sorting also explains things like variegated leaves in plants,
Yeah.
right? Patches of cells with defective chloroplasts.
Exactly, mitotic segregation of organelles.
So this leads to a big question. Why do they even keep their own DNA?
Yeah.
If the nucleus makes most of the proteins anyway, and the organelle DNA is mutation-prone, why not just transfer all the genes?
That is the million-dollar question in organelle evolution. Why maintain these separate, costly, potentially dangerous genetic systems?
Any good guesses?
Several hypotheses. Maybe some core proteins, especially very hydrophobic membrane proteins for electron transport, are just too difficult to import efficiently if made in the cytoplasm. Maybe it allows for more rapid local control over energy production. Or maybe it's just an evolutionary process that isn't finished yet. It's still a really active area of research and debate.
It's truly fascinating. What an incredible journey we've taken today, diving into these tiny powerhouses. From that universal concept, chemiosmosis,
Right. Linking membrane transport and energy.
to the specifics of mitochondria burning fuel and chloroplasts capturing sunlight, and that ATP synthase, thinking about it as a literal spinning motor inside our cells.
Turning over kilograms of ATP for you every day, it really brings it home, doesn't it? How these fundamental processes, conserved over billions of years from ancient bacteria, are still working nonstop inside every one of us.
It's mind-blowing, and it makes you wonder, what other secrets are these organelles holding about how life works, where it came from, and where it might be going?
Absolutely. There's always more to discover.
Well, thank you for joining us for this deep dive today. It's been fantastic exploring this with you all. We really appreciate you being part of our last-minute lecture family.